wonderful biology champions. I hope my dearest children are doing really great. Now, if at all you are someone who is super worried about biology examination and thinking that how can I complete the entire syllabus in one go and things like that, please understand, you now have just few days, and if at all you try to complete everything, it could be difficult to do that. And basically you might be shifting your focus on something that is not so important. So my dear children, make sure that you prioritize what is important in terms of marks. As I always say, do not think that I have not completed these many chapters. It's not just about the number of chapters, it's also about the weightage of it, right? So if you are someone who has time, go ahead, at least solve questions related to the topics that I am going to show you over here in your PU1 and also your PU2. Especially you have to focus more on your PU2. Complete PU2 as much as you can. If you have time, focus on the topics that I'm going to show you over here for PU1. And if you just want to watch the PU2 thing, you can just slide that particular video throughout. And if you think that this video had helped you, definitely let me know in the comment section that yes, it had helped you or not. I have actually put in so much efforts into finding the details. I've went question by question to give you something that could be useful for you guys, right? So let's make the best use of it, and do not forget to hit the subscribe button as well. So when you talk about first PUC, you have these many chapters out there. First, these chapters are something that you need to prioritize. I'll be showing you the order. We are going order by order only, so you need not worry. The first chapter that we have is cell cycle and cell division. Out of it, if at all we see in meiosis, okay, recombinase related questions, phases of meiosis 1 related questions, the pichitin bivalent related questions. There are 7 questions that have come from this throughout these many years. So this is a topic that probably you can go and cover. Nothing. Meiosis, in that meiosis prophase 1. In that prophase 1, you know leptotin, lazy zebra play during day. I always say these phases which are there, what is the use of recombinase enzymes in this particular phase and things like that. Question related to that can have high chance to come, so do not miss it. Okay, at least you solve one question, it will be understood. It will be easy for you. Yeah. Interphase G0 phase 1 question related has come only one. Mitosis related question has come one. So we never know what question could be coming in, but why to take a chance when you can have an opportunity? So do not miss out on studying these things. In my live lectures, in every lecture that I do, I try to explain to you. Okay, so make sure you solve the question. Take the PYQs. Do not attempt mock if you think that your PYQs are not strong. PYQs is something that gives you understanding of the concepts. Okay, so make sure you solve the PYQs first if you have got time. You can use PYQs as mock test, as in select the questions and give yourself a timer to solve those questions. If you have not completed a syllabus, if you have, then you can go ahead. Now, cell the unit of life. This is highly unpredictable, which is why if you think that you do not have time, it's okay to even skip it off. Why? If you have time, just cover the PYQs is something that you can. I'm talking skipping off PYQs is not what I'm talking about. PYQs you have to do. I'm talking about the entire chapter. If you're planning to go through the entire chapter, if you have not studied it, and if you do not have time, I'll not recommend to do that. PYQs, anyway, you have to do the PYQs. You will see that questions have come from each of these topics. So it is entirely different. When you solve the PYQs, and by chance, if you are lucky enough, if any of it repeats, okay, the concept repeats, you will get your marks. That is why I'm saying only 8 PYQs will be there, not too much. Okay, so you can invest your time in solving PYQs. Do not go away from PYQs. But I am saying that it is highly unpredictable. I cannot say what could be coming in, as I said over here. Okay, so here meiosis prophase 1 is important, means it is important. There are chances, high chances for it to come. High chances for it to come, but here I cannot give you a guarantee. But anyway, solve the PYQs for all the chapters that I'm going to show you. PYQs must. Animal kingdom also not predictable, because every year, every single topic, new things have been coming up. But please solve the PYQs. Only these many PYQs are there that you have to solve. Please solve them anyhow. Okay, don't leave them. Now, yeah, so yes. What about body fluids and circulation? Here something is there. For example, leukocytes, the highest leukocyte is neutrophil, the lowest is basophil. Basophil is the one that releases serotonin and histamine. This related question have come, 4 questions have come, so probably, again, this is a topic that could be helping you well. So whenever you are solving PYQ and when you are seeing this particular concept, make sure you go through it nicely. ECG related questions, 2 of them have come. So ECG also something that you can probably go through once, that particular topic. cardiac output, there's a formula for that, stroke volume into the heartbeats per minute. Go through it. You have got 2 questions related to it, so just go through that concept. This will hardly take 5 minutes of your time. It will not take too much of your time. So go through this concept, solve the PYQs of this, solve the PYQs of this particular chapter. Erythroblastosis foetus, 1 question, and lubed up sound and SAN node and everything, 1 question, and blood vessels also 1 question has come. So we never know what could be coming in, but at least these 3 topics are something that you should not be missing. Please solve the PYQs, all of it. Please, I'm telling you again and again, I know that could be irritating, but yes. Yeah. Now, plant kingdom, Baba, mostly unique features of the plant kingdom. For example, you know, protonema. This is something that is unique in the case of bryophytes. Okay. Microsporophylls, or basically double fertilization. All these things are very unique to different plant groups. All the unique things that you might know, please go through it once. Okay. That could be really helping you. 3 questions have come from there. Algae, Rhodophyceae especially. In algae, Rhodophyceae especially, such as, what do you call their reproduction, the storage pigments which are there, okay, phycoerythrin, fluoridine starch, all of these things have come, and Phyophyceae related 1 example has also come. So 3 questions have come from there. So these 2 topics are something that you should not be missing out. Again, while you solve the PYQ question, you will be able to understand it. If not, you are not able to go through the entire concept. When you solve the PYQ, you will know what is the unique feature. Okay, for Rhodophyceae and all, you know, in your NCERT there is a box for everything. Rhodophyceae, Phyophyceae comparison is there. Okay, go through that box and just check through. It could be helpful for you. Yeah, I'm talking about the scenario where you are planning to live the chapter by whole. Now, morphology of flowering plants, the flowers, zygomorphic flowers, example of biosection. Okay, apart from that, the floral diagram is something that could be coming to twice. There were questions that came from Brassicaceae and Solanaceae. So just go through the floral formula and things like that. If you're lucky enough, you'll get a question from there. Apart from it, everything else have come once. Estivation, position of gynoecium. You will see that when you solve the PYQs. As I said, the PYQs are very less. If you solve 10-year PYQs, only handful of PYQs are there, solve it up. Okay, you will get an understanding of it. Cool, I hope that works. Similarly, position of the gynoecium, epigynous, everything. So once if you see, you will get an idea of it. Don't live the topic. It will not take much of your time. Now, anatomy of flowering plants, arrangement of vascular bundles, Baba. 3 questions have come. This is a topic. This is a picture that is there. If at all you see an arrangement like this, where xylem and phloem are together, you call it conjoined. This is found in the case of stem, monocot, dicot, both of them. Okay, if at all you have a cambium in between, that means it will form secondary xylem, secondary phloem later on. This is a unique feature of dicot stem. If you do not have cambium, that is a unique feature of what? Monocot stem. When you have cambium, you say open arrangement is there, conjoined open. Okay. Open means open to change. You can remember like that. That is found in dicots. You're talking about root. Root will always have radial arrangement. If you have cambium, dicot root. If you do not have cambium, then monocot root. So that is the logic behind it. Just go through this particular thing entirely. It will be helpful for you. Yeah. So this is just an explanation of it. It will help you understand better. And yeah, I'll give you this PDF also. Neural control and coordination, these are the topics which are there. Again, quite unpredictable, but still midbrain is somewhere from where most questions have been coming in. If at all you can spend a little time reading about midbrain, it will be really good. And yes, that is the thing of neural control coordination. Biological classification, Baba, this is a thing. Kingdom Monera, mycoplasma. About mycoplasma, it's a living, smallest living organism out there, does not have cell wall, can survive in anaerobic condition. All of that is important. The shape of the bacteria related question, coccus, vibrio, spirillum, as things like that have come. Okay, 3 times it has come, so make sure you go through it as in mycoplasma, 2 and this one. So yeah, topic wise also have written. Kingdom fungi, groups and examples, twice questions have come, so make sure you go through that also once. And yes, rest everything is something that is unpredictable. It can be coming in, not coming in, yeah, like that. So biomolecules, again, these 4 things which are there, which are super important over here. One is this thing. Okay, some proteins and their functions, know about them, especially GLUT4. What does it do? It enables glucose transport. Insulin is a hormone, trypsin is an enzyme, collagen is something that is there in the middle. So please know about these things. You have what do you call the elements? The highest element, the one that is found in highest amount in the Earth's crust is oxygen, 46.6, that is the highest. Okay, now in the Earth's crust, the lowest amount is there for sulfur as well as carbon. Okay, sulfur and carbon. Sulfur is less over here in the human body also. also just 0.3. So these three things are super important: carbon, oxygen and sulfur. So just go through it once. Similarly, the Vmax curve of enzyme, and similarly the activation energy and things like that. If at all you have enzyme, you need less activation energy. If at all you do not have enzyme, there will be more activation energy needed for conversion from substrate to product. Okay. So about that it is there. So I have given you how many times the question have come. From table 9.5, thrice the questions have come. Okay, and yes, from table 9.1 once. So just go through it once. And structural organization in animals, over here just frog is there, so make sure you go through if you have time. I will not recommend you to go through the entire channel. If you have time, just go through some short notes, just read through. One question is only there, so you can just solve it. And yes, rest everything is deleted portion, so don't try to solve the previous year questions which are deleted, okay, from the deleted portions. So if you can go through a very short one-shot video, or probably just read the short notes if you have time, or else skip. Okay, now photosynthesis in higher plants, over here photosynthesis itself, source of oxygen and use of, where do you get oxygen from breakdown of H2O. What is the use of CO2? For the formation of glucose. Know that logic. Calvin cycle, what are the enzyme, what are the steps which are there. ATP production, what do you call it, chemoosmotic theory, and what happens, where it happens, and things like that. 4 questions. Okay, so depending on the topic it has come like that. C4 cycle, super important. Okay, where does the carbon fixation happens? In mesophyll and in the bundle sheet. In mesophyll you have pep carboxylase. In the case of bundle sheet you have what, rubisco. So all of this thing, events, Kranz anatomy, everything is important. Thrice questions have come. This is a very important topic. Do not miss it. Do not miss it by chance. If at all you get it, it will be a good thing, right? So yes, these 2 topics are there. Just go through it. While you solve the PYQs, you will get an idea. And yeah. And respiration in plants, again TCA cycle, acetyl CoA, oxaloacetate coming, the number of carbons which is formed per compound citrate is 6 carbon, alpha ketoglutarate is 5 carbon, succinate, malate, oxaloacetate is 4 carbon compound. So again just go through the cycle, TCA cycle once, it will be helpful. Respiratory coefficient, this is a very easy giving marks thing. Okay, 2 questions have come from this. Respiratory coefficient for fat is always going to be, if at all I have told you always. Okay, respiratory coefficient of fat, ulta of F, it looks like 0.7. Okay, respiratory coefficient of proteins, ulta of P, it looks like 0.9. Of carbs, it is unity, it is just 1. What is the formula? Amount of CO2 evolved divided by oxygen consumed. Okay, so in respiration oxygen is consumed and CO2 is being evolved. So CO2 at the top and oxygen at the bottom. And this is the thing, do not miss it up, very easy. If you feel that I am talking in Arabic, it's because you do not know it, but will you see the question, it will become super easy for you. So do not miss out on these things. Similarly, glycolysis, the number of 1 question have come and all of it, but do not miss out this. This is very important topics which are there in tool. I'm not saying that you have to miss the other options. No, you have to go through it as well. I'm saying go through it all, but please prioritize these things. Okay, so questions have come, which is why they are put over here, right? Yes, or else they would not have put. Now, plant growth and development, again, auxin, what is the use of it, apical dominance, and general feature of all the hormones, okay. Gibberellin, they are responsible for malting and bolting, okay, malting as well as bolting, 2 things are there. Over here, I have given you the most important things for all of it, so please go through these things. It could be really helpful for you to get an idea about the important role of hormones, yeah. So yes, and to the PYQs, as I said, 5 PYQs might be there for you to solve. Just solve it. It could be helpful. Now, excretory products and elimination, not predictable because multiple questions have come from multiple areas. But if you can, go ahead. If you do not, it's okay. Okay, but at least solve the PYQs, not go ahead with the much understanding of it. Living world, again, hierarchy of classification. King Peter came over for a great soup. Kingdom, phylum, class, order, family, genus, species. They can ask you the hierarchy in ascending order, in descending order, so please know that. Okay, and general matching order, you have to match the correct order and things like that. Binomial nomenclature related question, twice it has come. So yes, these 3 topics are there somewhere where you can score marks, so do not miss out on this. It's a very easy thing. Okay, the rules of binomial nomenclature and everything, who gave, Carlos Linnaeus gave, and things like that. Locomotion and movement, only sarcomere related questions have come. If you know this, you know everything. Okay, this is super important. Myosin is there in between, and towards the 2 sides you have actin, they come together, A band remains as such, the I band get reduced. This picture, everything working about this is super important. Just go through it. Thrice the question have come from this. Okay. Now, chemical coordination and integration not predictable. Multiple topics have come, but still, if at all you want to give a chance, just go through any short notes which are there already in the web. Also, you can just check for the short note mind map and stuff like that. If you get an idea of it, it's good. If something comes, it could be helpful. Yeah? Yes. So that is all for the first PUC chapters. From breathing and exchange of gases, nothing has come which would you have used. So next thing that is there is second PUC biology, and over here the first chapter, principles of inheritance and variation. Most questions have come from Mendelian disorders and chromosomal disorders. In Mendelian, as you see, color blindness, numerical questions could be coming in, sickle cell anemia related question could be coming in, or there can be questions where both of these disorders are being clubbed up and you have to match the following. So please make sure that these topics, Mendelian and chromosomal disorders, the questions are being dealt with nicely. You solve the PYQs nicely. These are all PYQs, but yes, just to have an idea of it. Okay, Mendel's pea plant, it is monohybrid, dihybrid, cross and everything. 6 questions have come from there. ABO, blood group. Okay, blood group plus pleiotropy. This is an important concept. Do not miss it out. Sex determination in general. Okay, generic way, 3 of them are there. Honeybee specific, only 1 has come. Pleiotropy specifically, separately I put. Pedigree analysis, symbol related, 2 has come. A disease, okay, you know, pedigree chart is there. Disease based, 1 has come. So just go through it. I'm just telling you what is the important areas that you have to revise. Okay, revise nicely. Genetic numericals, number of gametes formed when this is there, conditions like that. Over here you have to apply 2 raised to the power n formula. N means number of heterozygotes. So over here this is a heterozygote, this is a heterozygote, this is a heterozygote. So basically 2 raised to the power 3. Okay, so that is how you're going to do. Linkage and recombination 1, gene mapping 1, but anyways go through it. Polygyny related 1 question has come, test cross related 1, incomplete dominance related 1. So all of these topics are something that you should not be missing out by any chance, because from these topics questions have come until now. So go through these topics for sure, and then only go if at all you want to grab your 6 out of 6 marks or 5 out of 5 marks from this particular chapter. Okay, all of them, and prioritize this. I'm saying all of them you have to go through anyhow. PU2, there is nothing to live. Molecular base of inheritance, again these are the topics. From transcription, most of the questions have come. From types of RNA, that is the RNA polymerase is what I mean. Okay, RNA polymerase, monocystronic and polycystronic, post-transcription module splicing and things like that. In prokaryotes is different, eukaryotes is different. Okay, complementary strand formation, you might be asked to make a complementary strand. So how do you do that? 13 number of questions have come from transcription related topics are there. Okay, translation, UT or untranslated region questions have come. Amino acids, the form, okay, that has been come. Complementary tRNA, which is there, which is the complementary tRNA which will be there. You might be asked about that. Structure of the tRNA, amino acylation, all of it, 7 questions have come. Apart from it, genetic code. This is 6 questions have come. In the live of top 100, I have told you a way to remember the genetic code. If at all it could be helpful, check it out. Okay, there are 2 unique amino acid, tryptophan and methionine, which has only 1 codon. For methionine, it's AUG. For tryptophan, it is UGG. Apart from that, stop codons are also there. Yes, just have an understanding of it. Semi-conservative replication alone, this topic alone has given 5 questions. Lac-apron alone, 5 questions. Structure of DNA, the features which are there, and the structure of nucleosome, 4. Okay, intro to genetic material, that is genomic content in different organisms. Discover related questions again. The name of the discoverers. DNA fingerprinting, 3 questions. Human Genome Project, 2 questions. Griffith experiment, 2 questions. And DNA replication, 1. So do not miss out on any of these topics as well. Very, 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 very important. All of it. But yes, while you do solve more questions related to this, if at all you're getting confused between this thing, make sure you clear it up. Now, sexual reproduction in flowering plants, again, these are the topics which are there. Female reproductive organ is something that has most questions. You might be asked to calculate the ploidy in the number of cells. You might be talking about double fertilization. You might be asked about the embryo sac structure, where what is happening in the chalazial end. Antipodal cells are there. Degeneration of the 3 non-functional megaspores happens up there. All of it, super important. Okay, 11 questions have come from there. Pollination, clistogamous, chasmogamous flowers, autogamy related 1, water related 1, generic, where everything is being consumed. 3 of them have come. 6 questions have come from there. Now, male reproductive organ, that is how many pollen grains are going to be produced, if this is the number of anther which is there. The structure of microsporangium, that is epidermis and all the layers which are there, should know what is the function of the layers, what is the unique tapetum, has multiple nuclei, binucleate condition, it is having a dense cytoplasm, all of it. 5 questions. The male gametophyte, pollen grain structure, super important, alone gave 5 questions. Okay, seed and seed structure and importance is there. Structure as an embryo, seed, how do the labeling and stuff like that, especially monocot. Upper mixtures, polyamron and parthenocarpy. You have matched the following questions, 2 of them. Outbreeding devices, 1. Artificial hybridization, 1. Pollen pistil interaction, 1. Embryo monocot embryo structure, 1. So when I say individually a topic is giving 5 questions, that means that it is very, very important. So not just go by that particular number. There a topic is there, multiply it is divided, right? So yes, this does not make it less important. Go through all this thing. Now, human health and diseases, these are the topics which is there. From diseases, general disease, common cold, twice questions have come. Malaria 5 times, pneumonia 2 times, and generic 4 times. Generic as in where every information of disease is important. The first part of the chapter, no, it's very important. 13 questions. Drugs, generic 2, as in everything included 2. Opioids 2, cannabinoids once, and coca-alkaloids twice it has come. 7 questions have come from there. Cancer, carcinogen once, properties of cancer, what do you call metastasis and stuff like that, very important. Malignant property of cancer. Diagnostics 2, 6 questions. Types of immunity, generic, it is 2, all of them together. Active and passive and vaccination, 3 of them are done. So basically 5. So you need to understand this concept, then only you will be able to do it. HIV AIDS alone gave 4 questions. Allergy gave 3 questions alone. And anabolic steroids effect on females, 1 question. Okay, this is for human health and diseases. And finally, human reproduction. These are the topics which are there. Male reproductive system, that is the first one. Accessory ducts is something that has been asked most. Generic things are also coming where every information is being utilized. 6 questions. Female reproductive system, as I said, all these things are very, very important. Make sure you go through it. It's equally important. Hormones, placental hormones, thrice it is being asked. Generic, the name of the hormone, generally all of them together, once for 4. Gestation, the embryonic stages which are there, morula, blastula and stuff like that, and also month by month data, what happens in each month, 3. Thrice questions have come, 4. Spermatogenesis process and the structure of sperm twice, so basically 4 questions. Oogenesis process once, generic about it once, and also what do you say, number of gametes in comparison between female and male. So male, basically 4 gametes are being produced by 1 division, and there only 1. So 4:1 ratios. Please know that. Menstruation hormones which are there in implantation, progesterone is important for implantation. The date of ovulation, when will ovulation happen. Generic things related to everything clumped up together, 3. So mammary gland alone gave 2 questions. Parturition alone gave 2 questions. Introduction to events, as in first what comes, gametogenesis, and then transfer, okay, all of it, all the processes which are there in the introduction part of the chapter, please know about it as well. These are the important things. Now, reproductive health, contraceptives, natural, thrice it is being asked. Emergency 1, surgical 1, side effects once, and generic, as in all of them together, once. So basically 8 times, but most of it has taken from natural. So make sure you go through all of it, but give importance to this. Art alone gave 6 questions. Artificial, what do you call, assisted reproductive technologies alone gave 6 questions. Very, very important. Okay. MTP alone gave 3 questions. Amniocentesis alone gave 2 questions. Introduction to the chapter, family planning in which year, 1951. RCH program, what is the full form of RCH? Reproductive and child health care, all of it. Okay. So 2 questions have come from there. Now, biotechnology principle and processes, these are the topics which are there. A most important restriction enzyme, their naming and their source, their function, the restriction site, where do they cut, all of it. They can give you the diagram, they can give you the 5-3 direction, everything. You have to be sure that you are doing correctly. 8 questions have come from there, very important topic. Okay, gel electrophoresis, how is the naming being done? They can give you this particular enzyme is there, from which organism will you get this organism, this particular enzyme. So you have to know the logic, how is the enzyme named. Okay, so gel electrophoresis, elution, gel electrophoresis, generally 5 questions are there. From that, elution also gave 1 question. So vectors, PBR 322, 2 times questions have come. Selectable markers in the vectors, that is also super important. Total, if at all you see, 5 questions have come from that PBR and selectable markers. Are you able to select the transformants or whatever or not? So please know what is the definition of transformants, how are they selected, on all of it. Okay, insertional activation. PCR alone, Baba, it has given 5 questions, generic 2. Enzyme and the nature and the source, steps 1. Use and amplification of very less DNA, I hope you know that. PCR is something that you use to amplify the DNA if it is of very less quantity. Okay, so those related questions. Vectorless methods has thrice, okay, and that is a alone topic, so that is an important thing. Vectorless methods, microinjection and all of it. isolation of DNA, enzymes such as chitinase and lysozyme and all of it, okay, cellulase. So 3 questions have come from there. And generic, prokaryotes and eukaryote difference because of introns. So this can be there for the other chapter also, but it has been put over here. So since introns is not there, basically it is kind of different. In the case of prokaryotes, downstream processing, bioreactors, all of it clumped up together, you have got a question, generic question. Plasmid, the features of plasmid alone gave 1 question. They might be given which is not a feature of plasmid, so you should know. Now, microbes in human welfare, first important thing is microbe in industrial use. So generic, you have got 1 streptokinase rated 1, statin rated 1, cyclosporin rated 1. All these things are important. Now, chemicals, the other chemicals which are there, for example, Aspergillus niger is used for production of citric acid. Aspergillus niger is what? It is basically a fungi. So you should know who among them are producing fungi, who among them are basically bacteria. Streptokinase is the one that is produced by bacteria, streptococcus. Just all of them basically fungi. So you should know about all of this. Lipase used in oily removal stains, pectinase and protease used for bottle juices and stuff like that. Antibiotic rated 1. So basically 6 questions have come from there. Sewage treatment alone very important. BOD specific 2 questions, flock related 1 question, primary treatment 1, and generic all of it together 1. So basically 5 questions. So microbes in household again very important. And I have given you the terms also. Biogas production generic 1, methanogens related 1, IARI, that is Indian Agricultural Research Institute, and Khadi Village and Industrial Commission. They are the one who produced, who discovered the biogas plant and stuff like that. So you should be knowing about that also. 3 questions, okay, in total, all of them 1. Biocontrol agents, generic 2 questions have come. So you should know trichoderma, polysporum is not just for that. Trichoderma is a biocontrol agent also. Please know about that also, okay, do not get confused. Biofertilizers, generic 1, and generic again something that is generally there as a topic where it includes all the concepts together. 1 questions have come, okay, question has come. Now evolution, these are the thing, Hardy-Weinberg principle, numerical related to, factors affecting it related to, basically 4 of them. Evolution of man, 4 questions have come from that topic alone, very important. General terms and examples like convergent evolution and examples and things like that, 2 questions. Stanley Miller experiment alone 2, adaptive radiation alone 2, evolution of plants 1, convergent and divergent evolution 1, and all of it, as I said, 1 is there. Please understand that adaptive radiation, if at all it is 2 adaptive radiations are happening, we say basically is example of convergent evolution. So know that basic things. Once you do the questions, you will be able to analyze it better, but still. So my dear children who are getting very tensed, I have done in my 15 days challenge videos, I have done chapter by chapter explaining you the questions. If you feel it's needed, go watch it. 2x speed is also fine, but know about it. Very important topics are there. Do not miss out on this. Yeah, biotechnology and application, these are the topics which are there. And RNA interference is something that has given 6 questions, so this is a very important topic alone. PCR generic, and for finding problems with, as I said, that is there in this chapter also, this is important. Transgenic animal uses, especially alpha-1 antitrypsin, alpha-lactalbumin, 3 questions. Bacillus thuringiensis, crygene, what is the use, type of the organisms, lepidopterans, who are those, okay, dipterans, who are those, coleopterans, who are those, you should know that. And general content 1, okay, genetically modified plants uses 1, and everything else 1, but you have got 1 question, so you should know about them, at least a bit about each of them. Insulin also very important topic, okay. Now, organism and population, these are the topics which are there. Most of the questions have come from parasitism, especially general questions, along where you include a lot of all the information. Yeah, brood parasitism, Cuscuta is also there. In competition also, Balanus, Thalamus, warblers, general questions have also come. Population density, numericals are there. Factors affecting it, natality, immigration, emigration, and all of it. Type of growth, logistic, and the formula. 2 questions have come from that itself. So that growth curve is important, logistic as well as the other one. Make sure you go through it. Predation, monarch butterfly, and generic term has come. Introduction to population features. What is the features of the population? 1 question. Mutualism 1. Population interaction, symbols might be given and you have to tell what is the interaction. Growth pyramids, which is there. Growth pyramid related question is there. So please make sure you go through it. So 1 means this question has come. It can anywhere become 2 anytime. So do not ignore it. Ecosystem, again, these are the topics which are there. Productivity is somewhere where most of the questions have come from this topic. 4 questions. Different type of productivity. The equation GPP minus R is equals to what? NPP. what is the thing available for the next organism, what do you call the rate of formation of what do you call a biological biomass. So basically it is productivity. If at all you talk about rate of production of biomass by photosynthesis, it is GPP. So small term differences there, go through it. Energy flow calculation and photosynthetically active radiation percentage, 2 to 10% of it is utilized by plants, not whole 10%. So you should know that, all of it. Decomposition, all the steps alone it gave 2 questions, so it is important. Introduction to ecosystem, that is important. Food chain, important. General standing crop and all related question is in there. Ecological pyramids, the limitations have come, that is one. These are topics which you should not be missing out. Yeah. And finally, biodiversity and conservation. Over here you see species area relationship curve has been asked so many times. 3 questions have come from there. Tropical and temperate regions, okay, equator to pole will increase, decrease and stuff like that. And biodiversity hotspots, their feature, the conservation percentage, 30% would be conserved if at all we have this. What are the features of biodiversity hotspots? So you should know that, okay, there is habitat loss in biodiversity hotspots, so that is a feature of biodiversity hotspot. There is endemic species and all of it. So 2 questions have come from there. Ex-situ conservation example, 2 questions have come from there. Types of diversity, genetic diversity, you know, different examples of diversity, genetic, species, ecological, so you should know about that. Percentage of organisms present, example in vertebrates, how many of them are there, which is highest. So fishes is the one that is highest, all of it. Insects in all vertebrates and invertebrates put together and all of it, the graph which is there. Earth Summit, 1 question. Reasons to conserve biodiversity, you know, narrowly utilitarian point of view and all of it, one question. So these are the topics which are there that you need to go through. Okay, and I think with that I complete everything that is there, that is important, and it took so long to go through it, and I think it will be highly useful for you, at least now when you have your examination. Make sure, since you do not have time, this will be useful for those who have time to revise these things effectively, and for those who do not have time, make sure you revise the things that are consecutively asked, and all the things, not go by numbers. As I said, probably these years these questions can be asked, right? So you have to be careful regarding that. So please be sure of that. I just wanted to tell you that. And also if you want a mock test, there is ABHYAS, you can enroll into it. The pricing for that is rupees 999. Yeah, so that is something that I had to tell you. And my dear children, if you have any doubts and you want to ask me, you can ask me. I am here for you guys. I just want to wish the best for you. May all of you conquer your dreams. And as I said, if you think that this video had helped you, definitely let me know in the comment section. Wishing you the best. Please take care of yourself. Bye bye.